Okay, thank you very much, Laura, and thanks everyone for coming. So, in keeping with this morning's theme of death and destruction, my charge today is to tell you how phytoplankton can kill you. But it sounds doom and gloom, but I think we all realise that the point is that the science that we're doing here at CEOAS, while it may not enable us to prevent low oxygen or ocean acidification or harmful algal blooms, it might help us mitigate their effects by being able to plan for the future. And in other words, to paraphrase Ronald Reagan, I'm from CEOAS and I'm here to help. So a little bit of background about the biology that I'll be talking about today. As Jack has already said, every year we have upwelling-induced blooms off the coast here. They're usually comprised mostly of diatoms, and they're very important to maintaining the productivity of our region. There are some organisms, certain species of phytoplankton that sometimes are components of those blooms that produce toxins, and this slide introduces two of them. On the left-hand side, we have the genus Strutton, which is what we call a pennate diatom. They're sort of little siliceous needles. And on the right-hand side, we have Alexandrium, which is a dinoflagellate. There are certain species of Strutton that, under the right conditions, produce domoic acid, which is a powerful neurotoxin. That toxin, domoic acid, can accumulate in fish and shellfish, such as razor clams and mussels. And if those are consumed by humans and if the levels are sufficiently high, they can lead to illness and death. Similarly, Alexandrium produces a toxin called saxitoxin, also a neurotoxin, which can accumulate in shellfish. I'm using razor clams as the example here because it's one of the most commonly collected in the Oregon coast. And again, if they're consumed by humans, can lead to illness and death. So just a little bit of background on these organisms. I just mentioned the toxins they produce. To help prevent human illness and death, the Oregon Department of Agriculture monitors shellfish along the coast for levels of these toxins and closes those beaches to harvesting if they exceed regulatory limits. In Oregon, the harvesting of razor clams is mostly a recreational industry, and so we might think that it's not economically important, but when people go to the coast and dig razor clams, they spend money, so it's important to those coastal communities. And in 2003, there was an extensive closure at Clatsop, which is the focus of the recreational harvest in Oregon, and it was estimated that cost $4.8 million to coastal communities. So it's significant, and it's even more so in areas of Washington where the intensity of collection is greater, and also the local tribes like the Quinault Nation have a extremely intense economic interest in that fishery. So the work that we're trying to do is to understand the ecology and the movement of these blooms, understand the environmental conditions that might enable these organisms to produce toxin. We've been using some of the data collected at the coast. We've also been combining that with satellite data, and we're also trying to understand what are the ecological environmental cues that might bring about toxin genesis. So this is a map of coastal Oregon showing the locations where the Oregon Department of Agriculture monitors for shellfish toxins. So there's about 20 sites along the coast, and they've been monitoring for in excess of 20 years for at least one of those toxins. So we've been using the occurrence of these toxins at the coast and trying to backtrack to the near-shore oceanographic conditions using satellite data to see what sort of environmental conditions might be conducive to the genesis of these blooms and the production of toxin, and then how those blooms interact with the coastal shellfish and the toxin accumulates in those shellfish. So we've used these data to identify hotspots, so regions along the coast that repeatedly experience high levels of toxin and closures of those shellfish collection regions. We've looked a little bit at the relationship between the occurrence of toxins and environmental cycles like El Nino and La Nina. And we've also been looking at seasonal trends, so how the seasonal cycle of upwelling is related to the onset of toxin accumulation in coastal shellfish. So until we were doing this work, the information regarding toxin accumulation in shellfish was sort of spread among a couple of different areas and not always accessible. And one of the things that we've tried to do is to provide this data to the public in a more easily accessible fashion and combine the toxin data with the oceanographic data so that we can start to understand what are the oceanographic processes that might be leading to toxin accumulation. And so here's an example of these In these symbols here, we've plotted the toxin concentration, so that's domoic acid and saxitoxin, and these symbols here are cell counts of these potentially toxic species that are collected by the Department of Fish and Wildlife. So these are all offset from the coast so as to reduce clutter, but basically these are the sites along the coast where we collect these data, and here I've put out a chlorophyll satellite image that shows the productivity of the coastal ocean. So we're starting to try and disseminate this data, combining both the ocean data and the coastal shellfish toxin data so that the public can see potential relationships between the two. So using these data, we've also looked at regions along the coast that repeatedly experience toxin accumulation. And I'll show this slide to sort of explain the complexities of trying to determine if a bloom is toxic or not. On the left-hand side here is some in-water data of domoic acid concentrations. These are water samples that were sampled for the presence of domoic acid during an intense diatom bloom in 1998. As Laura mentioned, in 98 I was a postdoc at Mbari, and there was widespread sea lion mortality due to this event. The sea lions were accumulating domoic acid, and like I said, it's a neurotoxin. They were wandering up on the steps of the institute completely disoriented, and there was many of them died, but some of them were rehabilitated. So this was a coast-wide event. I've cut it off here, but there was extremely high values down in California. On the right-hand side, I've plotted a satellite chlorophyll image for that corresponding period, and this illustrates the point that I was making earlier, that sometimes these organisms produce toxin and sometimes they don't, and sometimes the toxin-producing organisms are present and sometimes they're not, and we can't tell that from satellite data alone. Here we have, over Heceta Bank, a hotspot of domoic acid occurrence, and we do have some high chlorophyll over Heceta Bank, but then down around Cape Blanco we also have high chlorophyll values, but almost no or very low domoic acid concentration. So this highlights some of the complexities of trying to identify a toxic bloom from satellite data alone. So right now I would say we're at the point where we can use the satellite data to identify a bloom, but we're still relying on in situ sampling, usually from vessels, to go out and take water samples and measure the presence and concentration of toxins in those waters. So this is a map of Oregon showing the point that I was making about the regions that are identified as hotspots that repeatedly experience toxin accumulation. So along the coast here I've plotted the 20 locations that the Department of Ag samples for the presence of toxins in shellfish. And here, using the Department of Homeland Security colour scale here, I've plotted the regions that repeatedly experience closures. So these two areas here on Heceta Bank and right up by the Columbia River mouth, about 25% of the time are closed for the collection of shellfish because of the accumulation of toxins. And so one of the we have some theories about this as to why this might be so. Some of the at certain times of the year, the Columbia River provides nutrients to the coastal ocean that could help generate blooms and potentially toxic blooms. And on the next slide, I'll just talk a little bit about this Heceta Bank region and what might make it a site of toxin accumulation. So this is a map of Heceta Bank from a paper by Jack and others in 2005. And what it emphasises is that the bathymetry over the bank here causes the coastal jet in the summertime to separate from the coast, and under certain conditions the surface circulation can actually come back onto the coast. So I showed in the previous couple of slides that this is also a region of potentially elevated productivity. So you can imagine a scenario where a bloom develops over the bank here. If the conditions are right for making that bloom toxic, then this potential advection back onto the coast, and this is a map of chlorophyll here. This is a mechanism whereby this toxic bloom could interact with the coast, and the coastal shellfish could accumulate toxins if they were indeed present in these waters. And this idea of interaction with the coast is also demonstrated in this animation here. So on the left-hand side, this is 1998 chlorophyll data for coastal Oregon. So this is from the CEOAS satellite. And on the right-hand side here, we're going from the beginning to the end of 1998. In the black, I've plotted an upwelling index. So when the black dots are above this horizontal black line, upwelling is occurring. And when the black dots are below the line, downwelling is occurring. So the point I'm trying to make in this slide is that downwelling is helping to bring blooms onto the coast where they might interact with coastal shellfish. In red here are domoic acid concentrations, and anything above this red line is sort of the warning level where they would consider closing the beach due to harvesting. So we start out in the beginning of the year, we're in the winter time, so it's relatively low productivity. As we get into the upwelling period, sorry. Okay, so I'll just let it run. As we get into the upwelling period, we have high productivity along the coast. Then as we go into the downwelling time period, that bloom that was relatively extensive along the coast is brought onto the shore where it can interact with the shellfish, and you see this huge spike in domoic acid in razor clams at this location in the Clatsop Beach after this upwelling has generated a bloom and then advected it onto the coast. So the upwelling causes blooms. We see that every year. For the most part, those blooms are held offshore as long as upwelling is continuing. We seem to see the highest impact in coastal shellfish when conditions switch to downwelling and those toxic blooms are brought to the coast. And that's illustrated again on this plot here. This is again for that Clatsop region, and this is the data from '98 that I just showed. So you see the switch to downwelling causes a huge spike in domoic acid in those shellfish. But I show this slide to also illustrate another point in that sometimes and this is particularly true for razor clams and domoic acid, when they accumulate domoic acid, sometimes it takes a very long time for them to depurate that toxin down to safe levels, sometimes up to a year, and that's related to the point I was making at the beginning about sometimes these closures occur for a very long time, which has a significant economic impact on coastal communities. So by getting a better understanding of the ecological mechanisms that generate these toxic blooms, and by monitoring them with satellite data, we hope to provide not only an early warning as to when these blooms might interact with coastal shellfish, but also help to minimise the time that the coastal shellfish harvesting needs to be closed. So to conclude some of the highlights of our work so far, we've identified these hotspots, Heceta Bank and the Columbia River, where the occurrence of, or the frequent occurrence of toxin in shellfish seems to be related to some of the local oceanographic conditions. I showed that the transition to downwelling conditions seems to be critical for these blooms having an impact on the coastal shellfish. One thing that I didn't talk about at all is the dinoflagellate Alexandrium actually has a cyst stage where these cysts rest in the sediment after a bloom has occurred, and they can help to generate blooms in the following year. In the Gulf of Maine, they do routine cruises where they try to map the distribution of these cysts and use that as a predictive mechanism for blooms that might occur in the coming year. And they find thousands of cysts, per cubic centimetre of sediment. We've gone out and surveyed the Oregon coast and we've found 7 cysts. So one big question over the Alexandrian blooms right now is where are the source populations of these cysts, and we believe that some of them could be coming from the north. We are using satellite data to try and identify blooms, and but we're still relying on in situ sampling to confirm the toxicity of the bloom. And we're just starting now to accumulate enough in situ domoic acid data that we can start to develop logistic regression models that relate the processes such as, or characteristics such as physics and nutrient status to bloom toxicity. We're also using new satellite products, not just the existing ones that are disseminated, to try and track the blooms. And ideally, we'd like to, and we've talked about this a little, collaborate more with the physical oceanographic groups within CEOAS to use some of their model outputs to predict the trajectory of blooms. And so ideally in the future, and this has some relevance to Sarah's discussion of ocean observatories, we might have sensors that would be telling us about the nutrient status of the coastal ocean, and hopefully in the not too distant future, sensors that would actually measure the toxicity of phytoplankton blooms, so we could get an idea of where toxic blooms are occurring, then feed that information into predictive models and watch where those blooms might go to provide an early warning for coastal managers for shellfish harvesting. And I'll leave it there. Thank you very much.
Do we have any questions for Pete?
That saxitoxin, is that the one referred to in pictures and that is PSP?
Yes, it's the toxin. The question was, what is saxitoxin? It's I didn't mention the disorders that they cause, but saxitoxin causes paralytic shellfish poisoning, PSP, and domoic acid causes amnesic shellfish poisoning, ASP. Yeah.
Yeah, I live in Europe, and I just wonder if the the same phenomenon is occurring in the Mediterranean? Do you have any information?
Actually, I don't, but I would say that in general, the US West Coast, the species that I mentioned are the ones of main concern. On the US East Coast, there are different species. In Florida, there's almost just one species that they're mostly concerned with. So in different oceanographic environments, there's different species, but it is a worldwide phenomenon, and there's also some evidence that the occurrence of the blooms is increasing and their toxicity is increasing, and perhaps due to things like ballast water transport, that the geographical distribution of certain species is also spreading.
All my life I've heard of PSP, but domoic acid was something that came on the scene, to my knowledge, only about 20 or 30 years ago. Is it really new here? Or?
Yeah, it's when I mentioned the Department of Ag, it's they've monitored for saxitoxin from 1979, but it's only since about 94 that they've monitored for domoic acid. So it's I guess it's something that we didn't really have an appreciation for until about the last 10 or 20 years. Yeah.
Are harmful algal blooms a very
Sorry.
modern phenomenon, or is there historical record?
Okay, it's an interesting anecdote I guess I could relay there. Is the Lewis and Clark diaries are all online now, and I've searched them to see if there's any mention of them getting sick from eating shellfish, and there's not. So no harmful algal blooms then, lots now. That's obviously they're increasing, but it's also true that I don't think they liked fish, so they didn't eat any shellfish. So, but there is evidence that their occurrence, frequency, and their persistence and their toxicity is increasing. But that could be just because we're looking harder. I better cut it off. Yeah. Thank you.
Thank you.